Hey biologist, Mr. Fa here. In this video, we're going to be talking about the endomembrane system. First up are the must-knows. Describe the mechanisms that organisms use to transport large molecules across the plasma membrane. So, as you can imagine, this is going to be all about how we transport things into and out of our cells. So, as I mentioned in class, this is one of the most important key functional things of a cell because it's going to discern the difference between inside and outside. Therefore, we're going to have to spend some time talking about, well, how do we get things either inside the cell or vice versa if we're to go from internal into outside the cell? How do things get exported? And that is in part where the endomembrane system is going to touch base. So we did not cover super in depth about our different endoplasmic reticulums, the Golgi apparatus, in part because this is where I more want to go so in depth to talk about the actual functions of these within a process, thus the endomembrane system process. So basically, at its nutshell, at its core here, rather, it's going to be about regulating protein traffic. So as I keep mentioning, proteins are a huge part about what this course is going to be covering and talking about because proteins are a huge part of life. They're a part of us and help what makes us us. So not just about trafficking, things entering and exiting, but also performing different metabolic functions. So if we do not happen to have this endomembrane system, we cannot get these proteins either inside or out of a cell to carry out specific functions. So it's kind of important because proteins are kind of important. So let's talk about it. So first up, the whole process of this is going to begin with the endoplasmic reticulum, thus the endomembrane system, right? Endo. So there are two different types of ER we're going to use for short. We either have the rough ER or the smooth ER. And the easy way to distinguish the two is one's going to have the ribosomes, thus the rough. Again, imagine that you are running your hand along like the surface, nice flat surface, and you felt bumps. It's rough. Well, what are the bumps or what are the rough bits? Those are going to be the ribosomes. And then if it happens to be smooth ER, it has a lack of ribosomes. Now, if we think of what the function of a ribosome is, it's to synthesize or to create our proteins. So a rough ER is specifically going to be dealing with proteins, whereas our smooth ER is not going to be about proteins as we look at the functionalities. So rough ER, the function, is really to package proteins for secretion, as we call it, or the export of these proteins to move from inside to outside. We're also going to be sending transport vesicles, specifically to the Golgi apparatus that we'll touch base on here in just a moment, as well as make replacement membranes. So as you can imagine, that things over time are going to get degraded, they get damaged, and they need to be repaired, and that is where our rough ER can then help us within this process. Smooth ER, notice again, lack of ribosomes, so not dealing with proteins in any way, shape, or form. Instead, its function is going to synthesize, which means to make or to create lipids specifically. It's going to metabolize our carbs, so it's going to be helping in the digestion process and the transport process of our carbohydrates. Also talking about detoxing of drugs and poisons. Additionally, about storage of calcium molecules so that we can use them for our osteoclasts and osteoblasts for the construction and degradation of bones. Now, those are kind of overall the general structure and function of each of the endoplasmic reticulum, but this process, this whole ER system, this is just the start of this. And so it depends on where it's going to begin. We can see two different images here. We at the top have our traditional cartoony looking image, where it's nice and neat and everything's organized. And then down below, we can see we have the more realistic or the scanning electron image that we can see down below. So just so we know that things are fairly familiar. And if you're ever given any of these images on an AP exam or any of our unit tests, I will identify, as they've also done in this image, if they happen to be too grainy. So I'm not trying to confuse you. AP's not trying to confuse you. So they will do their best to try and identify what are the different structures as to what we need you to get to look at to answer questions. So we can see its location is right outside of, kind of almost attached to that nucleus. So that nucleus is going to contain all of our genetic material, the DNA, and then it's going to help us start the protein production process. After it leaves through the nuclear envelope, the code, as we'll talk about later in protein synthesis, the mRNA, that messenger, it's going to make its first stop at an ER. Now, if it's going to be for protein synthesis, such as this example, we're going to head over to the rough ER. However, if it needs to do other functionalities like lipid synthesis or detoxing, as we mentioned before, then we're going to simply stop at the smooth ER instead. But rough ER is typically the first point because typically things are about protein productions. So it's going to get produced at the rough ER, and then it's going to create, when it's all said and done, was referred to as a transport vesicle. And so this is just kind of, it's like a little ball of our phospholipids. You may recall when we backed up last week and talked about our phospholipids, like the micelles. It's kind of like one of those, where it has the hydrophilic heads on the outside and the hydrophobic tails on the inside, and it forms this kind of little bubble, if you will, that it's going to transport, and we call this a transport vesicle. So that's what that one is referring to. This transport vesicle could then be shipped off to the Golgi apparatus. A quick and easy way to kind of remember what the Golgi apparatus is it's kind of like the shipping and receiving center of the cell. And so things can come inside, they can be the receiving side, or they can be getting shipped out, and so like the export side. So part of its functionality is to synthesize and package each of our materials, so things like small molecules, either for transport within the vesicles, and then additionally with production of our lysosomes, which are to break things down, thus getting into the whole detoxing and removal of harmful substances. They are very similar in structure to the endoplasmic reticulum, and they kind of look like these foldy looking structures, but typically what they do is, in the more cartoony looking versions, they try to use different colors. So you can see in the last slide that it's more of like a deeper blue, so we can see purple at its core for the nucleus, and then we have like a deeper blue for our endoplasmic reticulum, and now we have like a softer blue, kind of teal, moving into green hues. So they try to use colors to discern the difference between these. However, another way you can tell is that notice how we're moving away from the nucleus. We'll get to see it a little bit better in some pictures coming up, but as you move away, it typically goes in the fashion nucleus to ER to Golgi, and then to export. So if things are moving in an outside fashion, so it's exporting something, that is the progression. If it's moving internal, then it will work in the opposite direction, from the cell membrane into the Golgi apparatus, and then into the ER, and then it's going to go from there. So Golgi apparatus, again, is about shipping and receiving. It has two different faces, sometimes they refer to them as, and they have cis face and they have trans face. So this cis and trans are more so to identify whether something is going to be moving internal, so it's receiving, or it's going to be external, it's going to be shipping out. So typically, if it's going to be like concave, so it's going to be moving in an inward fashion, almost like a little hole. So this red arrow right in the center of this, where it has like an inward facing hole, that would be the receiving portion, and that would be the cis face. Versus the trans face, then it's going to be pushing in an outward direction. That's more for the exporting portion of it. So as I mentioned, here is kind of the pathway, if you will, if you were to follow the arrows. So moving from our core, that's the nucleus, having all of our genetic material, then it's going to move into the endoplasmic reticulum. Specifically, most of the examples we'll be talking about the rough ER because they have the ribosomes to do the protein synthesis. After it's done creating the protein, it will then package it using a transport vesicle. It will then ship it over, here's that transport vesicle in the center with the red arrows, into our Golgi apparatus. Again, moving from a core, from the center to an outward fashion. That's how we can identify this, as well as the coloration. Get absorbed into the Golgi apparatus, where it will then package and then remodify them. So if it needs to make any sort of adjustments either to the protein or the shipping transport vesicle, either or, it can make the necessary adjustments and then create an additional transport vesicle. Then it can push it into either an external side to move towards the plasma membrane to then remove it from the cell, or it can make it into an internal side in which it can then stay inside of the cell. It depends on what is going to need to be the function of this protein. You need to ship it to another cell, or do you need it for yourself? This is a great animation I found that basically just walks through and tracks the process from the core, that's the purple center, as it follows the protein production and moving in an outward fashion. I hope this helped everybody. Let me know if you have any additional questions. Thank you all for watching.